To the bsac weekly seminar. We have a special guest today. I think some of you know Roger Howe, but maybe you might not. So he is actually one of the, wasn't on his bio here, but I think if I'm not mistaken, it's one of the original faculty at bsac.
Not correct.
Oh, okay.
Well, I was here when it was called BISC, Berkeley Integrated Sensor Center, and the leaders were senior professors, Richard Muller and the late Dick White. And then two young guys showed up, one a little before me, Al Pisano was a young assistant professor. And then I moved from MIT as an assistant professor, and we became the assistant directors to the full directors. I was close, but.
Okay, well, anyway, so he is currently an emeritus professor at Stanford, although he is still teaching a class there. And he got his PhD and master's here at Berkeley, as you mentioned. And then he had faculty positions at CMU and MIT before coming back to Berkeley, where he taught for 18 years. So he was here for a number of years and was, as he said, bsac director back in the days. His research group at Stanford has been focused on nanoscale design and fabrication for a variety of applications. He was the faculty director for the Stanford NanoFab for a number of years, and also was the director of NSF's National Nanotechnology Infrastructure Network. He co-founded Probius to commercialize research from this group on broad spectrum biomolecular sensing. And we welcome him back to Berkeley today. And thank you for coming.
Okay, thank you, John. Whom I know well since he was at Stanford until he came across the bay to be the executive director of bsac. He was Systemex's executive director. So I have a talk, and is my audio working well? Can you tell? My ex-PhD students is in the audience on Zoom. So, okay, well, the title is on this quantum electrochemical spectroscopy, digging into some neat academic research that then led to a company. But I've cut that down a bit. Why? Because the back end of the talk will be about an academic career. And I think that's very important. You're sort of in an academic career, those who are not on the faculty. Now, there may be faculty members in the audience that are of a different generation. No? Okay. Once I ran into a professor at Stanford. I thought she was a senior grad student or postdoc, but introduced herself as a professor. So sometimes that happened to me early in my career. Down in 231 Corey, I'm looking for grad applications or something. What are you doing in here, right? And well, I'm looking for potential grad students. So I'm going to go through this technical talk, which I think is interesting to those who have backgrounds in quantum mechanics. Might be a little interesting. I'm not going to get into a whole lot of that. There's some pretty hardcore circuit design. There's also some interesting challenges in fabrication, both in the university and otherwise. But I'm going to go also present some things that have not been presented yet on this startup company that has Berkeley people involved, of course. Counting myself, but there's also others. And then this whole issue of an academic career is something that I was asked to give a talk last week at the University of Florida on that topic. They have a series of talks because they have people getting PhDs who may end up in universities. So I'm giving a very abbreviated and subset of that that's directed at you. So how do we detect molecules in the assays that you give blood, maybe you, or you get a blood sample and they do this panel and they're measuring all kinds of things? Well, there are different ways to do it. This is a common one that leans heavily on our friends in chemistry. College of Chemistry is a huge asset to Berkeley, both chemistry and chem engineering. They design chemicals that can bind to surfaces to be a probe, and then label chemicals that can bind to the analyte that fits the probe and be lit up in at a wavelength. And anyone in the audience know what HRP stands for? Horseradish peroxidase. Is that weird? Who knew, right? That pretty nasty tasting stuff actually can be very useful in biology modified somewhat. Well, we can easily, all of us in the room can build a detector for the light that we can excite a fluorescence at a convenient wavelength, and we get a signal and we produce the result. If you want to detect another chemical, you get the opportunity to take your friends in the College of Chemistry out to lunch again and ask them to design a chemical to do that. And each one is a laser focus, and that's got advantages. What's the other way to do it? Well, I'm going to talk about inelastic tunneling spectroscopy that few have heard of, but some have heard of Raman spectroscopy, mass spectroscopy. All of these are broad spectrum techniques. Put the sample in and you get all the information, and then you're looking for fingerprints. Well, what is IETS? This is adapted from a figure in 1968 at Ford Motor Company. They're doing basic science research. They accidentally discovered that the pump oil, they used diffusion pumps in those days, got in between these metal layers, and that they were seeing some interesting signatures in the IV curve. Okay, that's interesting. And in fact, it's still used. This is IBM Zurich work a few years ago where they're identifying the different bonds from the signatures when they're sweeping basically an IV curve. Though if you look closely, this is a second derivative. These guys were just using the conductance. This is accentuating the peaks. We're getting beautiful signals. So this looks great. Why isn't it used more widely? Because the fingerprints are really nice at 4 Kelvin. Okay, so bring your liquid helium, right? It's not something that's going to be super convenient to do. So what at research in my group, collaboration with Boris Merman, who is a bsac alumnus, until recently a professor in my department, a circuit design guru, and Ron Davis in the medical school, we had a project to work on a phenomenon observed by a student at the University of Illinois who became my postdoc. And I'll just run through this to give you the idea. You have the soup, sometimes called the matrix, the chemical information. Think of serum or some other biological sample, whole blood. Sample of peanut flour, if you're looking, that might be mixed into wheat flour, any sort of mass information. We're going to try to capture it all. We're going to get an electronic readout and digital filters and quotes. It's a lot of machine learning, and get a detection result if we've trained the system to find the particular molecule. Because we're going to get, as you'll see, a tremendous amount of information from the sample. And basically, we're going to be looking for signatures in the current, and this is going to be at room temperature. And that's going to take some explaining. How can we do this quantum stuff washed out at 300 Kelvin? So one thing that's interesting is I'm an EE, and so the communication with my chemi postdoc is challenging. My goal, actually, and his advisor's goal, was to pull him closer to EE and nanofab and to launch his academic career. This brilliant University of Illinois PhD student who joined my group, it failed. He's not a professor. He's CTO of a company at this point. But in any case, talking about interface charges, classical and quantum, doesn't make sense until you get a circuit model. Okay, so this is a paper that we co-authored where we're trying to figure out what kind of interface at the working electrode is going to give us that information. And it turned out that a self-assembled monolayer, which are these linker molecules, how many of you have done HMDS vapor treatment? Great, okay. So you've done a kind of self-assembled monolayer to promote adhesion to the photoresist to the surface. So anyway, the chemists have designed a lot of these. We can get them in different lengths. This thickness matters. The insulator surrounding it, the area of the sensor and the parasitics. And ultimately, you can derive, if you do a long paper in the Journal of Physical Chemistry C, you can figure out estimates anyway of the monolayer thickness, the sensor to parasitic area ratio, and the redox chemistry in the soup. We're actually going to dilute the sample into something like a ferricyanate redox couple that will allow current to pass. So, okay, what does the sensor look like? Well, the work at Stanford, we ended up binding a gold nanoparticle to the electrode. It's kind of a funny figure. As you'll see, it reflects the funny way we were making it. This is platinum iridium. The area of this sensor site is around 50 by 50 nanometers. Okay, if it's too big or actually extremely small, that's also a problem. Remember, we're doing all this modeling to get the kind of appropriate range. And there's a linker to the nanoparticle and some methylene groups on the outside. And we have this redox species that will allow current, because we are measuring current in this. So what do? I emphasize we are doing nanotechnology. Well, we work hard on a circuit. Boris Merman, Ross Walker was a student. He was a professor at the University of Utah. Left. I don't know if you knew Ross. It may have been a bit before time. Well, this is a patent on something that is interesting. It's a, remember Clark, where we were increasing the damping on a resonator by electrical feedback. This is essentially pulling the damping out, but it's not a classical system. It is a quantum system that we're doing it to, hence the mesoscale system feedback induced. Welcome, Professor Piester. So in any case, we have that symbolically here. We have some JFETs for noise considerations and a feedback loop, the goal of which is to get really low noise electronics, kind of record low noise. And the circuit looks like a potentiostat. These are used by electrochemists all the time. And in fact, we made a board level implementation, but it's a pretty good potentiostat because I bought one of these up here. This is the power spectral density of the voltage noise. Spent $10,000 on that one. And this is Boris Merman's one. So he learned a lot from his advisor, Bernard Boser, who's emeritus from Berkeley. Boris actually left Stanford as a professor at the University of Hawaii now. So anyway, it was necessary to do that. And another student of Boris's, Sean Fisher, ended up putting most of it onto a chip. So you have a CMOS chip that is the interface with a lot of other extraneous stuff because we're putting samples into a well and we have our working electrode here. But a huge attention to shielding because, quick question. Now, what week are you on at Berkeley? Week 4. Okay, we're at 5. Okay, so it's midterm time, right? Or it should be midterm time. Don't have midterms anymore. Anyway, no, you're a semester school, so life is good. Okay, you're actually learning something, and the professors are actually not grading exams or what other miserable stuff. We're halfway through our quarter, the winter quarter. Big problem. But anyway, what are these spikes? Pardon? So you can, yeah, 60 hertz. We're surrounded by that, though some of these, you wonder with the LED revolution, it may be going down, these interferers, right, that can be filtered out, that there's 60 and 120 and maybe some other squiggles that are always leaking in. So we would do tremendous shielding, and you will in your thesis as well. Okay, so lots of people do potentiostats. And this is ISSCC and Journal of Solid State Circuits. People make giant numbers of channels. You know, they're doing arrays. We're not doing that. We have 3. The power we could care less about. The area is a don't care. It's the voltage and current current floor and voltage noise that really matter. And these are quite respectable, though not necessarily best of all, but the scan is extremely slow. We're going to sweep the voltage over 30 minutes, and we'd like to go faster, but some of you may have had circuits courses in 1/f noise, and the problem of going slow with electronics, people modulate to get away from DC. Even Clark's resonators are noisy as you get down to really, really low frequency. So anyway, this is the circuit side. One interesting thing is we would put that Gamry that I spent $10,000 out of the contract on the electrodes, and then we would put the custom ultra low noise. We would get quantum information out of the Boris Merman group designed potentiostat and not out of the other one. So it does matter a lot. Last topic, and I've tossed out a lot of slides to make room for the end of this talk. One thing is people say, what is the effective temperature of the electrons when they're tunneling? And it's about 20 Kelvin, because we have sucked the noise out of that. Now, temperature and noise are tightly related. And so remember, the IETS was a 4 Kelvin measurement, but it's only the electrons that are at 20 Kelvin. We'd like to go colder than that. So how do you make a sensor? We decided to use the sharpest substrate. People complain about curved substrates or rough silicon or something. This is a needle. This is a scanning tunneling microscope tip made out of platinum iridium. I've deposited a ALD layer of hafnia. I've punched a hole with a focused ion beam and done my chemistry on the tip. Why on earth did we do this craziness? Well, I had a postdoc, Shuai Chang, who got his PhD at Arizona State. The whole world for him, it really didn't exist if it wasn't on a tip. And he convinced me that it was cheaper and faster to prototype chemistries serially in the beginning, and then later go to substrates. So I said, okay, go for it. And so there is a STM tip, and there's the hole in it. I actually had undergrads punching the holes. We would have this thing that looked like a pin cushion, and in the focused ion beam, you'd go punch those holes, and sometimes they would punch a whole thing like target practice of holes on the end of the tip. So it was fun. How do you get them into something that's going to be sealed? Well, one idea is to pull them backwards through our friend PDMS, that silicone that we use. And there is a tip that's on its way to getting pulled perfectly. And it can go down just right. In order to do this, grad students are not very helpful. Why? Too much coffee, too much Brownian motion in the hands. Okay, so I went to high school students to do this, and they were really good. No coffee. Then, of course, they end up going to undergrad and end up getting the jitters. So anyway, our DARPA project was looking for BONT-A. What is BONT-A? Some people inject something like it. This is a surrogate for the nerve toxin, but you've heard of Botox? People get old and they get wrinkled, and then they kind of get injections that make their faces look kind of wooden and frozen because they're paralyzed. This is the stuff. And what we had was something safe for even EEs. So I had spent a third of this big project over 3 years with some great work on sample preparation, because even mass specs have sample preparation, right? And so I thought my postdoc was more enthusiastic, but I thought that we should probably make sure that we get the abundant proteins out, as much of the junk out of the sample, so that we could get a cleaner measurement. Even if it's broad spectrum, you can't just put it in the solution. So what came out of that funding? Some of you may know Sam Emiminejad is a professor at UCLA, and Mehdi Javanmard is a professor at Rutgers. So 2 EE PhDs in this project ended up going the academic route. Jeff Rogers was the program manager, and at the time, he's the one with a bunch of, we're the only bio project in this kind of quantum sensing thing. Most of them were physics optics peoples. And he just said, stick that thing in serum. You claim it's broad spectrum, see what you get. So we did. And you get a lot of interesting IV curves. These are the actual curves with different amounts starting at 100 attomolar, okay, up to picomolars of these IV curves. And my PhD was a chemical sensor. And if I showed this to Richard Muller, my advisor, he would he would be not amused, because how can you interpret that in 1984? What he was hoping for is something that looked kind of like chemical sensors often have a rise time and, you know, exponential and then decay, and everything is nice. This is horribly ugly. So what do? Well, we differentiate it like the IETS people do to accentuate the peaks. And we're essentially looking, you think of the IV curve, what if you get a channel? Okay, what are the physics? Well, one of these biomolecules ends up on the electrode. How does it get there? There's a current going through this. We're running current through the electrolyte, and so it's actually dragged there, okay? And it ends up there, and there's current flowing, and when the vibrational electronic states of the molecule are lined up, you get a tunneling current. So you get steps in current that then turn into peaks in the so-called conductance spectrogram. How do you analyze the signature? Remember, this has everything in serum. The background is ugly enough, and then you add these. Well, the strategy is we're going to have the sample and a couple of references. We're going to find out what that bond A looks like in buffer, right? That's a classic clean solution. And then we're going to put the buffer in the analyte together, and we'll have a negative control of pure serum. So we're going to do some fairly fancy, but actually rather mundane signal processing. So to quantify that, what do? You've had wavelets. Some of you are EEs, taking signal processing courses. De-embed the SAM. The SAM is that thing that's on the sensor. If we have no analyte, we still get a signal because we have organic molecules on the sensor that are helping with the tunneling. So we de-embed that. We strip it out. And then look at independent components of the bond A reference, independent components of the serum spike, do some cross correlations, and we get an assay. I'm deleting a slide because I want to make room at the end. Remember, it's a little bit of a fast tour. And I gave this talk almost exactly 4 years ago. Who were here other than the faculty? And you're a faculty member? Okay. So you were a first year and now you're a fourth? Almost? Okay. But you remember this perfectly. No, I don't think so. Anyway, but what's neat is that ELISA is this enzyme-linked assay that's often done, and it's not seeing anything at the kind of detection levels we're able to reach. So that's really encouraging. The DARPA people said, wow, we were right, it worked. You didn't believe in your own technology. You get this ribbing. Then a week later, after discussing it with some people, I said, well, what this means is there isn't anything like bond A in your blood. Okay, it kind of stands out, this neurotoxin. If we had blood samples, there would be 0 in all of our blood because you'd be dead otherwise, right? So it's this nasty thing. So then he said, go find some cytokines, these signaling molecules. So we were able to do that using slightly different things. So I want to go through the commercial side because I have not been able to talk about this much until I got permission, from the higher ups, meaning the CEO and CTO, both of them former postdocs of mine, that we are out there selling stuff and we can talk about things. So this Probius company, I'm going to talk a little bit about what it's up to commercializing this. So I had a spinoff out of Berkeley called Silicon Clocks that ran for a few years, was acquired, and that was a silicon germanium resonator on top of CMOS, so really pure EE. This, I'm leaning on my chemi minor. Are there any chemies in the audience other than the faculty? Bill, you're a chemi, right? and a bona fide chemie. Okay, I took 3 classes from like Clay Radke and Harvey Blanche, right? Yeah, anyway, and so these were killer classes, absolute, you know, the chemies, you don't, in fact, yesterday at the EE faculty meeting, we were astonished at how the chemies were doing their faculty search. It was unbelievable amount of work, okay? And we just don't work like chemies. We work pretty hard. I was on the weekend going through these folders and all to try to find out who to do Zoom interviews with, right? So one thing, if you ever apply to a university and PhD UC Berkeley, you know, we get hundreds of applications, okay? Many of them, and you'll see this hierarchy business, but Berkeley is one where you go to the top, right? And then there's people reading, like 5 professors for every folder in our department. And the chemies, they're, you know, contacting their kindergarten teachers or whatever. They're, you know, it's over the top. So anyway, what have we been up to? Well, we were founded in 2016, coming from Stanford. We've raised money, including 5 million. What does this non-dilutive mean? It means from DARPA and other, or NIH. In our case, it was DARPA funding before the company. And then the program manager said, he's not going to fund any more academic work. You guys got to do a company, basically. So we got some injection at the beginning, and we've raised other money. Total is 11 of the VC money. And we have 24 patents and patent families and everything. And Stanford OTL, just like Berkeley OTL, will figure out a way to get the company going rather than license it to some big, there's seats in the front, come on up. Historically, the people that really invented this and their friends that get going in a company are the ones who are going to really drive it commercially, and not some monster biotech company. So they actually, none of those were interested. At Stanford, they would have to, for due diligence, they advertised it, but no one was going to license the patents, thankfully. And so we got the licenses, and we have continued to patent stuff. So basically, it's this QES is the secret sauce. Applications are too broad. Okay, the latest DARPA funding, last bit, was for this disease. It's not a disease, it's a condition called sepsis. It's a blood infection that's very deadly and that can kill people of all ages. It is a very serious thing, and it's very difficult to diagnose because kids come in with a flu, kids come in with this, but if it's sepsis, you want to treat them radically differently and right now. So anyway, my dream is to live long enough to see it in an emergency room for this triage purpose. And so there's some interesting bioprocessing quality control. I think I'll talk about insulin, different types of insulin. We have an instrument, and we have consumables or disposables, okay? Kurt Peterson once said, that's the dream company. As people throw out your product, it doesn't get any better. They have to buy more. But of course, what they throw out is very inexpensive, right? Because otherwise they will go broke. So, you know, platforms, you know, the usual stuff, software as a service libraries. What is the software? The analytical tools to pull the information out. And some interesting things. It's not here, but I like the word a posteriori assays, because you gather all the information. If you want to look for something else, you don't have to get more blood. If you have a digital twin, if you've heard that, of the sample, and you then proceed to do an additional reference outside, you know, and then you go look for something new. I'll look at that. So we've detected single cells. What kind of cells? Like salmonella is something that causes food poisoning. Gave a paper at a food poisoning conference. We're focused on proteins. We're not very interested in the DNA side, but even small molecules like putting a deuterium on a instead of a hydrogen amino acid, we can detect that. So how do you detect that? Because of the difference in the tunneling, right? I mean, it's a different vibrational frequency, different energy, and we're able to pick that up, assuming you're interested in looking for such things. That was kind of so that's the subatomic thing. That's not really interesting, but the big thing is very little blood sample phenotypes, right? That idea of getting a broader indication of what's going on rather than like 3 markers. Does this new drug damage the liver? Is not answered by 3 markers. There's a global response that we think we can capture. So here's what the thing looks like. And you can even see a video. I'm not going to be one of these guys that stands here with a video, but you can go to our website, probius.bio, and look at that. So the QES disposable vial is shipped with the electrolyte in it. Okay, so we have a seal, and then the user punctures the seal with a pipette tip or something to put the sample in. There's no reagents, no sample preparation, small volume. Put in a pipette, put in the sample in the 30-minute scan. We keep working on making that shorter, but compared to a lot of assays, which may be hours or days, that's pretty good. We have a huge amount of effort of working on what amounts to machine learning algorithms to more efficiently pull out information. And so it's an interesting business that would, we're trying to classify, identify, correlate, quantify, all of this stuff. And this time travel is where you can go look for new things that you didn't even know you were interested in. So you're all fab people. How many of you have been in the Marvell Nano Lab, one of the best on the entire planet? Raise your hands. Okay, I guess I was in the predecessor that was with Professor Wu. We overlapped a little bit in the 20th century, doing some hydrofluoric acid with Q-tips, right? Not approved currently, but in those days it was very useful. So there's a front end here. There are chips, right, with small pores. I'll go through what that looks like, but you're familiar with fabrication. We thin the wafers. We do ENIG, electroless nickel immersion gold, and build up some pads, dice it up. Then we go through flex fabrication, clean up the sensor, depanel it, and ultimately we have a separate track to do the vials. The gold pins are inserted. They're gold coated, not solid gold, for reference electrodes. And the whole thing is laminated. And of course, you can't read that. So the next slide, I'm blowing things up. But the big picture is a little bit different than classical MEMS. But your MEMS, even if you're in the pristine resonator end of bsac, those are going to end up packages, right? And that's incredibly important for to make something that is useful. So how many of you heard of NextFlex? Professor Piester has. This is one of the US manufacturing institutes started in the Obama administration. The first one was in robotics in Ohio in maybe 2014 or something. This is one of the last ones that has continued. It's headquartered in San Jose, but they have kind of sites all around the country. What they have is a development lab for flex electronics that potentially, I think, Berkeley partnered with Flex in some way. Students could appear there. Your PhD could be accelerated. You could get out of Berkeley and be in San Jose for a day, which I did on multiple occasions anyway. It's sometimes worthwhile to break things up, especially if the people you're meeting are going to do something that is difficult to do in the resources at Berkeley. So look them up, they were of tremendous help for us, because being in Fremont, we actually would be down there and working with them to develop processes. Now, this is not flexible electronics. This is a nanosensor in a foundry, and it's not as hard as MEMS because it's the back end of line of CMOS. 3 metal layers. Here is the sensor pore, which is not focused ion beamed. It is deep UV lithography. And if you look really close, you can look over there. There's Hafnia sidewalls that choke it down to about 75 nanometers. So it's a little on the big side, okay, but it works. And it's nice to not have E-beam or especially focused ion beam to be drilling away. So there's lots of metal and there's lots of shielding on this, a lot of attention to coupling of signals and that kind of thing. But this isn't rocket science, I mean, this is familiar to you. But then it gets interesting. I hadn't ever looked at flex panels, and this is what happens when the chips end up getting mounted into these little tabs that are made of PET base coverlay with silver ink in between. So they have machines to do this, pick and place machines to put the chips on the, it's kind of like cell phone components, right? There's a lot of automation in that, but not a lot of that in the universities, and laser drilling, and we end up with our flex circuit. But then that has to go on the vial. This vial has 4 of them. And if you look closely, there are 4, there's a division of walls between where each sensor is looking. And those don't have a label on them. There's no super specificity, but we found that the N group of the SAM, there can be different interactions with different classes of molecules, whether it's an alcohol or a methylene or something. So we've played around with different SAMs, but some of these we just do with one. So how do you do vials? Well, it's precision injection molding, putting gold pins in. That barcode was not trivial, it's very small. And a lot of engineering that would be not as familiar, okay, to me anyway, but you can have this stuff done. And then you get to the software. I have one paper, one of my last PhD students got hooked up in doing analytics for this class of sensor, and she developed some new things in collaboration with a statistics and math prof. But in our case, I'm just going to give you the 30,000 foot view. You have, if you have interleukin 6, you do serial dilution in something like buffer. You may actually head off any interferers by doing some off-target analytes to get their signatures. And that may be done computationally, not in vitro. You go dilute the target analyte into the matrix, and then you're doing all of these scans. And you identify what you're looking for is the analyte-specific vibrational signature. There's all of those signatures. How do you pull that out? Well, you've got a lot of information on what it should look like, and then you end up adding this. Every one of these is adding to the model database. So you're building up this software that ultimately then can address. So these are references. Here's our blind samples at the end. So there's a lot of run up to get this show on the road. But once it gets going, you begin to build the data and you have something that could be quite valuable. For instance, you might go poking around in blood that I gave a year ago. If there's that might be of interest for my colleagues in the medical school who work on geriatrics, the study of aged humans, and there's some interesting thing with this. They could go get another data point without poking my arm, which is good. I don't actually mind blood draws. Here's a funny story. We were raising money in the seed round and gave a talk, we meaning, I couldn't make it as Emmanuel and Chaitanya, the Sandhill Angels. And this is right when the Theranos scandal was erupting. And one of these VCs said, "How are you guys different from Elizabeth Holmes?" And, that's an interesting question. And Emmanuel said, "We didn't drop out." Just like you won't drop out. Yeah. So no, we got our PhDs, right? And she dropped out after 1 year as a fresh after the first year. So anyway, we went up to Toronto, and there's a lot of interest in food safety, and did quite well on a whole bunch of different types of things that you may be interested in looking for contaminants for food. That's interesting. Won an award for new technology. benchmarked it against the gold standards, but it's not our focus. What are we up to? Well, this is a collaboration with Merck. Merck, not that branch of Merck that is down in San Jose doing fab equipment, right? There's a huge company. This is the drug end of Merck, and they're looking at drug-induced liver injury in rats. This is a 4 microliter sample. And in fact, that is a very, very small sample. I'll just put in why are they interested in this? Well, they're getting a lot of new information, but they're also getting the fact that the rat doesn't have to be killed. You can pull a little bit of blood with a syringe out of the tail, and they go about their business, and you can follow them for the whole trial. Other assays, they have groups of rats, huge numbers of rats. They don't make it to the end. Every single sample, they end up taking a cohort out, literally. And so this has gotten some interest, fewer lab animals, more information. So in any event, this question is, can you do this panel? They're interested in a specific marker, and so we went ahead and did that against a Siemens machine. The Advia 1800 is what they is standard to use, and it's kind of scattered. We have a very tight correlation between these two markers, which is exactly what you would expect. And more interesting, these are these cascades of tests done in drug screening. And they said, well, what about inflammation? Well, these were measured after the fact using references without needing any more serum from the rats. So we captured that, trained new references, and then did the analytics to get the inflammatory response. Okay, my colleague Niaz Baini, MD in the pathology department of the medical school, was very interested in TB, tuberculosis, a huge killer. Problem is the WHO standard test is a high-tech nucleic acid amplification test that you need to have highly infectious samples of sputum or saliva, and if someone has to get them and be in a biosafety lab, and a lot of worse tuberculosis is that isn't the case. So can we do this from a tiny amount of serum? And the answer is we can certainly separate. This is this initial study in 2022. We have 10 positive, 10 negative samples. They're clustering very differently on this particular warm-up. But in fact, what we found out after the fact and had hints in the data is 13 of the 20 were HIV negative and 7 were positive. Okay, so can you go about identifying these people getting both TB and HIV? And it's not perfect, but it's very encouraging. So Niaz is getting more samples, and we're running these things. So we have our instrument in labs, in places like University of Colorado, Brigham and Women's Hospital is part of Harvard, and some companies as well that are digging away, trying to see this for different applications. So it's exciting. With that, I will have a few minutes at the end, but I'm going to go over. Is that allowed? Is there a class in here? Okay, because I think I spent a lot of work on this University of Florida talk on faculty careers. So they have a series of these. I was the second after Evelyn Howe of Harvard. So, I went way over on that one too, but they stayed because there was no class. So my background, I'm the son of an EE from Purdue who went to the Air Force because he was an ROTC, so you have to serve in the Air Force flying planes. And then in the late 50s, he went to work for SMUD, Sacramento Municipal Utilities District. And because he then power in Purdue. You know, power is something that was lost for a long time and is now in the upswing. But this was the beginning of transistors. So he went to UCLA and USC to take grad classes to learn about this. He never got the master's, but he took a ton of classes. One of them was from a grad student at Caltech named Richard Muller. So there was an intersection between my advisor and my dad when I was about 4 or 5. I remember him doing homework on the kitchen table. And so I went to Harvey Mudd because it didn't have a PhD program. I got into Berkeley in physics. And Berkeley was so huge. Harvey Mudd was so friendly. But what I didn't know, it was Harvey Mudd was hardcore. There was 144 of us and 88 finished. There's a picture of our class I couldn't find where they whited out all of the people that didn't make it. Might have lived happily ever after. They certainly got more sleep. Anyway, I came up to Berkeley. I was admitted to Stanford and to Berkeley, okay, and to USC and UCLA, but those were out because my future wife was a student at UC Davis, and the distance was too far from Harvey Mudd anyway. So anyway, I was going to go to the Bay Area, but I called up the Harvey Mudders, who, because we didn't email, I found their phone numbers and called them up and talked about what their experiences were at Stanford and Berkeley. Okay, in Berkeley, it was just tough. Really, really tough. The courses were hard, even by Harvey Mudd standards. And at Stanford, they were having a good time. Okay, the courses weren't that hard because they were quarter courses designed for industry. There was a lot of industry people taking them. And there was no thesis for the master's degree, whereas in those days in Berkeley did. And I didn't know, I didn't want to get a PhD, but I wanted to learn how to do stuff. So anyway, I went to Berkeley and noticed it's also a lot closer to Davis and there's no bridge. So I could get there fast. In those days, there wasn't as much traffic. So I surfaced at UC Davis. So I married after 1 year. I didn't want to get a PhD, wanted to return to HP Labs where I'd done a summer job. But what happened? Well, the economy was bad. If you think interest rates are bad now, they were like 15%. Okay, so it was just a wipeout. And I was a lab TA for EE 100 at the time, was a course for mechanical engineers, and I loved it. So if you wanted to teach, you have to have a PhD. Better sign up for the prelims, right? And those were not fun. But I talked this over with my dad, and he quoted me this saying, which you may have heard. Okay. I didn't know who said it. I didn't think my dad invented it. I didn't like it very much. But anyway, George Bernard Shaw is a famous Irish playwright. And I haven't seen the play Man and Superman written 120 years ago, but that's what it's from. Okay. Shaw was not a professor either. Anyway, so my dad was a VP of engineering. You've ever used a nanospec? Do you still have one around, the thickness gauge thing? Yeah, so he was working in Silicon Valley and had been at Fairchild and all. And he made this statement that microprocessors had just been introduced, okay? Just becoming real. And there was so much excitement going on. It had been so bad in the 70s and now things were going to explode. He was right. And so he said, why not leave with a master's and do something that will change the world? He's selling me. I'm going to waste my life as a professor, okay? And it's a good question. What would have happened? I often think about that, right? Our students who are not professors have done some amazing things, right? We lost a PhD student before I got to Stanford. His name is Zhensheng Wang. He left after, halfway through his PhD. He had this idea for this company, NVIDIA. And he's such a strong, determined guy. He just disappeared and made that company a success. It's sad. We've offered to have him come back, but he's awfully busy with his $1 trillion company and the resources of NVIDIA. So it's not, again, it's not for everyone. He made a huge impact, but he doesn't have a PhD. I'll remind him the next time I'll be at a safe distance. Anyway, so I went to Carnegie Mellon for a year. The clean room was delayed. I'm a creature of Marvell or the SNF. I couldn't imagine a life outside of that. I could have started a program in simulation, right? But I'm not that smart. So I leave and I go to MIT. Why? They have a lab that's open. It wasn't really fully up, But then I find out after 2 years of real exciting stuff at MIT that I get a call from my advisor. They got an NSF IUCRC. There's a new kind of research center with industry and all, and it's in MEMS. It's called BISC. So actually, we got a faculty position because of that center. Why don't you come out and interview? So I did, got the job. Now, coming back to a university with your old advisor there is interesting. We didn't collaborate particularly. We wrote some review papers for quite a few years, and then later on, it wasn't a big thing. I think that was not consciously the case. It wouldn't have affected my career. But actually, imagine coming back if Clark is your advisor or Ming, and then you have an office, in Sutarja Dai, and it says professor on it, and but then people remember you as a student. It's a little bit awkward. If you're at MIT, they don't know, they don't care. So it's easier to do that. But anyway, I got kind of run out of town by an organization that you may not know about. It's called the University Office of the University of California Office of the President in Oakland. Okay, there were 1,700 people there at the time, and they had a new patent policy change. I was the head of EE as Clark is, right? EE division. And I got the email and I read it. It was a big mistake. If I'd have just had an automatic spam filter for anything out of those guys, I would still be here. Yeah, it was that kind of thing. I fought them for 2 years. I heard from Liwei Lin that the faculty won 5 years after I was at Stanford. So at the age I was, I might not have survived another 5 years of war with these people. But anyway, I've been here at Stanford, there at Stanford for about the same time. I'm half-time now through March 2025. It's one of these academic early retirement incentive programs. I'm half-time in effort. I'm not half-time in pay. Anyway, so I'm easing into the pasture. Academia is not an egalitarian institution, right? You weren't selected by lottery, okay? We're going through grad admissions. The professors are obsessing and arguing over who's the better student, okay? You didn't know this? That's, it's serious stuff. Why? Because we absolutely need the absolute best students, okay? Now, what is best? We don't really know, right? The people actually change during the PhD, but this is a Nature paper. You've heard of that journal? This is about academic hiring and whether you go up or down. Almost everyone goes down, okay? What does that mean? You're at Berkeley, that's off scale, and you end up somewhere else. UCLA. Okay, I could Stanford, I won't say anyway. So in general, that's kind of not surprising, right? You're at one of these super top schools. There's not that much room up there. I was at the University of Florida. They've got a lot of very strong faculty there who went to very strong schools, and it varies, from all these different departments, from pharmacy to religious studies and all. And I think that it's nice to be in Berkeley. I gave this talk, the same slide in Florida, and Florida is not up there. Maybe someday, but it's not now. There's just a handful of schools that produce a huge number of professors. Why? Like Caltech's not on there because they don't have any students, okay? It's a tiny little place, right? MIT isn't on there because it also isn't as wired into all of academia as Berkeley and Stanford are, and Wisconsin and Michigan. So guess what? Your PhD opens doors. It opens doors with grenade kind of explosion, right? Believe me, it means a lot. But it's more complicated than that because all PhDs aren't equal. And the expectation now is to do a postdoc, right? And probably at another school, not necessarily. Maybe even in a different field, right? I had Chaitanya Gupta, this CTO of Probius. He came from Illinois as a chemie, okay? So, but his co-advisor was an ME. What you end up doing is having a lot of opportunity cost. You may have heard that in economics. Like you could be building a company. You could be going up the ranks where each promotion leads to step functions in salary, right? Down in Silicon Valley or wherever, right? You can do lots of different things, but no, you're working as a postdoc and the salary isn't particularly great, but maybe you get a chance to make a research breakthrough. And there are postdocs who are not interested in academics. They're interested in starting a company. A lot of Stanford postdocs come there, work hard, make a breakthrough, launch the company. So just as an aside, by the way, industrial experience is viewed very favorably in engineering. So you go off and do stuff in industry, we will find out about it. How do we find out about it? Networks of intelligence gathering by the universities, okay, that are competitive and overlap. If there's someone hidden somewhere, we try to find him or her and get them out and get them in front of a class, right? It's hard to find the best people. So I think the future is that you may end up going from industry to the university to the startup. Maybe you'll take a turn in Washington or another government running a research program, right? That's possible. When I left, I got good advice. Richard Muller said, you must go to where there are great students. So even if your office is a closet, even if the building is run down, if you have great students, it's wonderful. You better have a clean room. This, that's self-evident, right? If you don't have a clean room, oh, but there's this little detail. You need talented, committed staff, the right set of faculty tools. If the clean room was focused on something that you're not doing, then the dean is going to have to drop money on you or on the clean room in order for you to work there, or you're gonna have to change your research, right? So this is a big deal. And in my career, I had someone come up and say bluntly, get out of chemical sensors. Why? It was 1984. Notice that this Probius company is doing something with chemistry, but it's not 1984. You work in a field with high quality papers that have defined performance targets. That often means there's companies associated with them. And you're mixing in these IAB meetings with the leading people in the MEMS industry, right? Now, you don't want to do what they're doing, but to do something connected to what they're doing is important. Your advisors are helping with that. But chemical sensors, Richard Muller thought it would be great to get a chemical badge for clean room workers, because there was, what was the motivation for my project was preventing cancer in the lithography workers in Silicon Valley. Who were exposed to xylene because it was negative photoresist in those days, a lot of it, and that's a carcinogenic compound. And so my thesis was a xylene vapor sensor. So, in radiation, you wear a badge and they log your exposure to radiation. If you have too much, you get to rotate out. So you thought people would go in, get exposed, and then stay out for a month, and then that would be managed. But you know what the chemists did? They just got rid of xylene. And this was Simon Middlehook, who was one of the founders of Silicon Sensors. Bluntly, he visited Berkeley and he said, get out. And he was right. Okay, there are some, there's turbulence coming in many dimensions. Gerhard Casper was, the motto of Stanford is in German, Die Luft der Freiheit geht. It's like the wind of freedom goes, but the exact translation with my 1 year of reading German. Anyway, it isn't quite what they thought it meant, but it's sort of like the freedom of academia or something like that. But he was actually a German guy and came to be president of Stanford. And he gave a talk in 1996 that got noticed across academia because he said the US spends 0.25% of everything on genuine research and development. And even that amount, which our grad students rely on, is threatened. This is probably before you were born, right? So it's been a long time coming. Why is that? the US has a problem. Lots of countries have problems. Back in 1971, when I was in middle school, you had a huge amount of discretionary funding, and you had some mandatory funding or entitlements, and then a little bit of interest. And then a couple of years ago, it looks a little bit worse. And if you live off the discretionary funding, as we do, it looks really bad. What are the mandatory? Social Security and Medicare. If you listen to the two candidates, they are totally committed at age 77 and 80 that they will not cut Social Security, they will not cut Medicare. Fine, but the alternative is you better, you better jack up something like taxes or cuts. You got to deal with it. Now, Gerhard Casper is from Germany, where the budget's balanced much better than they do here. His criticism of Americans, fundamentally, they're not Germans. Or they're too happy, too optimistic. We'll always wriggle out of it, right? The doom is coming, which the Germans think is coming tomorrow every day. And okay. So anyway, so this, that's our research funding. And this is discretionary. I think we got a problem. So our research funding is going to have to decline. So what to do? Gerhard Casper launches a fundraising drive, not for buildings, but for graduate student fellowships. And to date, he's long gone, but there are 300 of them, and they've raised 200 other fellowships. So these are fellowships that put people off of the grant from NSF or DARPA or whatever. And in fact, Richard Muller's alums across the world, we all pitched in, and we have a graduate fellowship named after him. Which unfortunately is not a bsac person. So as I'm wrapping up, this is a book I read during the pandemic by Jeremy Bomberg, who people may know. He's at Cambridge, director of the Nanophotonics Center. It's called The Secret Life of Science. And it's about what it's like to be a professor in a research university, what it is like to manage the funding agencies, to manage your college, the rise of social media and what it means to our existence. So higher education is going into turbulence. I had a very rough ride back from Florida in that storm that hit a couple of days ago, and it's going to be very turbulent. Casper looking decades ahead, huge changes are coming. What's coming? Well, there are people at Stanford, chair of a humanities department at our long-range planning. Will this Stanford University even exist in the 60s? How can a university like Stanford disappear? Maybe if you're worried about it disappearing, it won't disappear. Okay, maybe you can do something, but maybe it used to be a horse farm for Mr. and Mrs. Stanford. Maybe it'll be a horse farm again. Universities cost a huge amount of money and they don't do a very good job. And what is the change coming? I think there's huge opportunities to disrupt the university system in a way that will be incredibly painful for some, incredibly good for the next generation. Steep declines in, now did I miss a slide? No, I didn't. It's the next slide. Anyway, do people have confidence in higher education? Not as much, and it's certainly been declining. I think these don't really work, the Apple and Meta goggles, the immersion goggles, but I think there's a way to do labs in your bedroom or at a community college that would be equivalent to the labs that I TA, okay, because they're realistic. If you don't plug in the instrument in the metaverse, it doesn't work. You can capture the whole thing, and people can get a running start in an engineering career coming from a much broader base of people. And I think there's the end of the time served model. What do I mean by that? People spend 4 years and they get a bachelor's degree. What does that mean? Well, increasingly, it doesn't mean a whole lot. Okay, so I think there's going to be what the British done, these exit exams that are where people are identified as qualified or not. And so the hard work that people at Berkeley will do will matter. The degree will be validated, and the schools that don't do well are going to have a problem. You may have heard of ChatGPT. It's disrupting Stanford. I'm sure it's disrupting Cal. So how do you have an exam? Have you ever seen a blue book? And they're blue at Stanford as well. And the pencil, right, is this invention, you know, and you can code on paper. Chat is not there, right? It can't help. And so you're toast. No, you actually have to learn the material. Actually, oral exams are another way to find out if you know what you're doing, because it's a little awkward to go on your phone and look for Chat GPT. There was an EE prof who was challenged because his answer didn't agree with Chat GPT. Circuit design is hard, but there's 2 Stanford students I've met launching different startups for AI-fueled co-pilots for circuit design. And was looking for a grandson in algebra. There's Conmigo, an AI-powered tutor that for $4 a month can help you learn, right, by finding out your weaknesses and all, just like your own TA. So all of this, I think, will be an I think the 21st century is going to be very exciting. And so all of you are potentially qualified to be professors. And but what does it mean to be a professor? What is education? In this huge turbulence, I've encouraged my students to maybe stand aside in industry and watch to see what the transformation is. I think there'll be universities that do survive in some form, but they won't be quite like they have been for the last 1,000 years. So with that, I thank you for staying here late and would be happy to discuss. I guess you eat lunch here, or how does it? No.
We're gonna have to run you over the fact that we're gonna shut the kitchen down.
Well,
unfortunately, we'll be able to take any questions.
Okay, no questions, because those are all tagged and they don't have my name on them, right? Okay, well, they need it much more than I. So with that, I will sign off. Thanks. Do I leave Zoom then? Okay.